Hello dear students, I am Dr. Ashish Agrawat. Welcome to my channel Live Biochemistry. In this video, we will discuss about the glucokinase versus hexokinase. As we know, glucokinase and hexokinase both mediate the same step, that it converts glucose to glucose 6-phosphate. So what is the difference between these two? So the first difference is in the location. Glucokinase is present in the liver and pancreatic beta cell, while hexokinase, it is present in all the tissue. In the liver, this glucokinase enzyme help in the glycogen synthesis from glucose. In pancreatic beta cell, there is a glucose sensor, so glucose will go inside the pancreatic beta cell and it will synthesize the ATP. The ATP that is produced will open the ATP sensitive potassium channel, and so that the voltage gated calcium channel will open and calcium will release the insulin. So this glucokinase enzyme is present in the liver and pancreatic beta cell only, while this hexokinase enzyme is present in all the tissue. Second difference is specificity. Glucokinase has a narrow specificity. It acts only on glucose, while hexokinase enzyme, it has a broad specificity. It will not only act on glucose, but it also acts on the other hexose like fructose, mannose. So hexokinase has a broad specificity. It will act on the glucose, fructose, mannose, all the hexose, while glucokinase will act only on the glucose. The third difference is affinity for the glucose. As we know, glucokinase has a low affinity for the glucose, while hexokinase has a high affinity for the glucose. So what is the advantage of this? Because glucokinase has a low affinity for the glucose, so it will only act when there is a sufficient amount of glucose is present. So after the meal, when there is a heavy influx of glucose, then this glucokinase enzyme will help to utilize the glucose in the liver and pancreatic beta cell, while this hexokinase has a high affinity for the glucose, so it can bind with the glucose even at the low concentration and facilitate the production of the ATP. The next difference is Km for the glucose. So the glucokinase has a high Km value because it has a low affinity. So Km for the glucose is around 10 millimolar, so it is around 180 milligram per deciliter. So whenever the glucose level is very high, then only the glucokinase will work, while hexokinase has a low Km value, so it can bind even at the low concentration of the glucose like 1.8 milligram per deciliter. So it can bind even at the low concentration of the glucose. As we know, the Km value and affinity, they have an inverse relationship with each other. The Km is a Michaelis constant, so it is a substrate concentration at half maximum velocity. As glucokinase has a low affinity for the glucose, to reach the half Vmax, it requires a higher Km value. So it means it requires a higher substrate concentration to reach the half max. So that's why the Km for the glucose, for the glucokinase is very high. Okay, it is around 10 millimolar, while in hexokinase it has a high affinity for the glucose, so it will bind with the glucose even at the low concentration. So the Km value is low because Km value is a substrate concentration at half maximum velocity. In case of glucokinase, we require the higher substrate concentration so that to reach the half maximum velocity. That's why Km for glucose, for glucokinase is very high, while Km for the glucose, for the hexokinase, it is very low. Okay, and inverse relation with the affinity for the glucose. So next is function. So as we have discussed, this glucokinase uses glucose only at the high concentration. So after the meal, okay, this glucokinase will work at the liver and pancreatic beta cell to regulate the influx of the glucose following the heavy meal. Okay, while hexokinase will have a function of basal metabolism of the glucose. So it will facilitate the generation of ATP even in the low glucose concentration, as it has a very high affinity for the glucose. So even at the lowest concentration of glucose, hexokinase will help conversion of glucose to glucose 6-phosphate, and then by glycolysis it produces the ATP. Okay. The next difference is reaction. So glucokinase, the reaction is rapid, so it is a high Vmax value, while in case of hexokinase, the reaction is slow, so it has a low Vmax. The sluggish reaction of hexokinase makes sure that only required amount of glucose is utilized. Next is feedback inhibition by glucose 6-phosphate. In case of glucokinase, it is not inhibited by the glucose 6-phosphate, while hexokinase is inhibited by the glucose 6-phosphate. Here the feedback inhibition by glucose 6-phosphate on hexokinase makes sure that there is no unnecessary glucose is utilized. The next difference is inducible by the glucose. So glucose substrate will itself induce the glucokinase enzyme and it will form glucose to glucose 6-phosphate in the liver and pancreatic beta cell. So the influx of the glucose is utilized after meal. While in case of hexokinase, the glucose will not induce the hexokinase enzyme. Okay. And the last difference is induction by insulin. So glucokinase is not induced by the insulin. Insulin will stimulate the hexokinase enzyme and by that it will stimulate the glycolysis process in all the tissue to facilitate the generation of ATP by glycolysis. This glucokinase enzyme, it is a type of hexokinase type 4. Okay. Well, when we talk about the hexokinase, it is hexokinase 1, 2 and 3. While hexokinase 4 is called as the glucokinase. I hope you understand the difference between glucokinase and hexokinase. If you have any doubt, please write it in the comment section. If you like this video, please hit the like button, share this video to your friends, subscribe to my channel so that you can get notification for my future video also. You can also visit my website www.livebiochemistry.com where you will get study material regarding the biochemistry. Thank you again for the watching. Bye.